I'm going to be kind of shifting gears for the talk, away from chemistry for a second to biology. As you may know, my name's Jason. I'm excited to talk about some of the new AI stuff Scilligence is working on. I've been working with one of my colleagues, Ray, to work on this really cool tool for ELN. So AI is everywhere now. You hear it every week, every day. ChatGPT, machine learning, all that fun stuff. And the lab's no different. AI is coming to the lab. It's going to be the future. And Scilligence is joining that craze and moving in with it. So the angle we took for this project is to kind of automate away the things that biologists don't really want to do and need to get done and be more accurate with it. Specifically when it comes to cells. With our tool, you'll be able to prep a sample as you normally would, upload an image to our ELN. Our model will analyze the sample image, and then it'll return in a few seconds the cell counts, cell labeled, with the overlay of the labeled cells, and then you can finalize the data. So save a bunch of time, increase your accuracy. And so as I kind of went over these steps right here, but then also, as I just mentioned, it'll minimize human errors in subjectivity when you're counting the cells. You don't have to have different lab techs or a scientist counting the cells in different numbers. They'll just be the same number with the model. You'll also free your time, as I just mentioned, so you can actually work on the actual science you want to do, not just sit at your bench and count cells all day. And another cool thing that's a little different that a lot of the other cell counters that are out there don't do is it'll also phenotypically catch cells that you may not have otherwise been looking for. And I'll get into that in a little bit. So I'm going to go through two use cases for this. The first one is going to be a complete blood count, which is done every day and around the world, thousands of times a day. It's just a general test to get to assess a sample to see how the specimen's doing. You can use blood count to get an idea of any sort of anemias that might be happening, any sort of blood infections, cancers. And so our model can do this, and can assist with this. So our model can get a quick, a few seconds count of red blood cells, white blood cells, and platelets to help you assess sample. Over here on the right side is an output from the model. As you see, it right there, it labeled all the cells it saw, gave a confidence interval or a proportion that the model was certain that was correctly labeled. And I'm going to go through in a second here an actual video of the workflow. So I'm just going to play this. So right here, we're in our Scilligence ELN, and we have our template here. We just named example cell model for this, and we're just going to add an image, upload it. The screen capture software I used didn't capture this other window for some reason, but I just selected a file for my downloads. There's our cell image we're going to use. Over here on the right column, or left column, sorry, you can label the sample, and also look to the counts in the model in a second. So right here is going to give us a generic name for your own housekeeping. You can do whatever you want to, whatever kind of system you want to name this as. We're going to get to the fun part here, where we're going to now select our model we want to use. Oh, we need to select the actual item. Yep. Now we do that. Now we're going to do the model. Again, for some reason, the screen recording software I used didn't have this drop-down menu, but there is one there. I just selected the blood cell model, and now it's going to have the model analyze the image. For this demo, this is still a dev environment, so it's going to take about 10-15 seconds. In production, this will be under 5 seconds, 3 seconds. Yep, so there it is. In a few seconds, we have the labeled image. We also have the cell counts. As I mentioned before, each of the boxes has a proportion, which is how confident the model was that the cell was properly labeled. Yep. And we're going to go over this one second. So kind of going over this. And what's kind of cool too, now we're going to, is you can now filter it. So say you wanted to only view, cells which were over a certain threshold. I think I used 0.8 here. Yep, 0.8. And you wanted to do only red blood cells. So this is going to do is it's only going to show cells for red blood cells and have a confidence of over 80%. So it helps you get a different look at different data. Visualize in different ways. We're going to do it again. So we're going to do 0 here, I think. Yep, 0. So this is just going to show all the white blood cells. There's the white blood cell. And I think we're going to do another one for the platelets. Yeah, right there's our little platelets. And then we're just going to show them all again. So just again, the complete blood test or blood count is run multiple times every week. It's really used throughout every day, and using a tool like this would speed up that process dramatically, save a bunch of time, increase your accuracy. Yeah. So now we're going to kind of switch gears to another use case. Oh, why is it? Oh, stop. Okay. Which another to do more of a phenotypic diagnosis. So instead of just getting an assessment of our sample, we would be using the phenotypes or morphology of the cells to catch pathologies. Yeah. So in this example, we're going to be looking through acute lymphoblastic leukemia. Excuse me. With these kind of things, time is one of the most important things in a medical setting. Time with cancer is probably one of the most important things you can have, is catching things early on. In a research environment, this can save you a bunch of money and resources if you have a sample that suddenly has cancer that you didn't intend to have cancer. Save you time and resources that way. It's also very labor intensive and error prone process, and so this kind of model would expedite that process, streamline it. What's also kind of cool is, right, you could use this in different ways. So you could even just not even use this to intentionally seek out leukemia. Let's say you could have this kind of as like a watchful second eye, where if you have someone that's trained on just blood cells, let's say, but not on leukemia, they might not notice that there's leukemia in this data set. So then the model could be used as kind of an alert system, where it's like, oh hey, you should go take a second look at this. There might be some abnormality. And as we'll get into a little bit, this is just an example. These past two examples, we can adapt this to all types of morphologies and phenotypic uses. These are just two. What's also kind of cool with this approach is the examples I've gone through have been about public domain data, which we've used to train our models, but we could also incorporate this with private data or specific patient data to create a very specified data set. We could train a model with particular patient clusters of lymphoblastic leukemia, make our model particular in a certain way that you want to stratify and find specific differences, and have clinically relevant output or results that we wanted to target. So again, this is kind of just a use case for it. There's endless uses for this in different ways. So again, I'm just going to show a quick recording of the lymphoblastic leukemia. It's a little shorter. It's not going to go into all the filtering. We just saw that in the last one, but it's the same kind of process. You just upload an image, and you choose from the models. You choose, instead of blood cells, we're just going to do the acute lymphoblastic leukemia data set. Okay. And again, this will be faster in production. Yeah, so right here in this data set, it's kind of binary. This will be different in use case when it's finalized, but in this case, the model either only tags them as all acute lymphoblastic leukemia or normal. This will be incorporated into the general blood data set, so it would be more specific with the red blood cells in the background. In this case, it was just tagging leukemia. Yep. And this kind of say this is the beginning, right? As I mentioned a few times, this is just a few use cases of it. We're going to be improving user control immediately with it. So you'll be able to, as you probably saw, there were some cells that were missed or maybe the bounding box wasn't perfectly around the cells, or maybe it labeled something wrong. And so the user will be able to go in afterwards with our filtering tool and add boxes, remove boxes, resize them. And so that way you can use this tool kind of as like a way of getting a quick rough draft or of the cell slide, and then you can have your scientist just finalize it. So it speeds up the process that way. We're also going to be expanding the models. We already have a few types of anemia in the pipeline. We have sickle cell anemia, an iron-based deficiency anemia, I think a malaria one as well. So a bunch of different types of phenotypic based diagnoses we're going to be expanding this to. And we can adapt this to client needs and requests. We're happy to work with you and figure out how we can apply this to your use cases. Just again, for the last time, this is just two examples of, it's just a cell count and leukemia, but you can imagine there's a bunch of different types of ways we can integrate this or use this throughout our applications. So yeah, that's all my talk. It was kind of brief. Yeah, I'll be excited to answer any questions anyone might have. And if not, I'll be passing this over to my colleague, Senna.